diagrammatic representation of recombinant DNA technology. Observe the diagram first. So what we require? We require foreign DNA. What do you mean by foreign DNA? For example, if you want to produce insulin, you require a gene, you require a DNA capable of producing insulin protein. So this is foreign DNA. Or if you want to produce any growth hormone, then this DNA capable of producing growth hormone. So foreign DNA, or you can call it as desired DNA. First, we have to cut the DNA into fragments. You can see the lengthy DNA is broken down into small fragments with the help of an enzyme, restriction enzyme. The enzyme name is restriction enzyme. Again, I'll explain. So we are taking foreign DNA, we are taking desired DNA. Desired DNA is cut into small pieces. That small pieces, you can observe this. So this action is done with the help of restriction enzyme. Now the question is, what is a restriction enzyme? See, in the year 1963, the two enzymes responsible for restricting the growth of bacteriophage. Bacteriophage is a virus. One of these added methyl groups to DNA while other cut the DNA. So the enzyme which cuts the DNA, we call it as restriction endonuclease. So restriction endonuclease is an enzyme which cuts the DNA. The first restriction enzyme is HIND2. Here HIN represents Haemophilus influenza. Functions depends on the specific DNA nucleotide, isolated and characterized 5 years later. It was found that this enzyme cut DNA molecule at particular point, at particular sequence of 6 base pairs. This sequence, we call it as recognition sequence. There are 900 restriction enzymes. Now we know more than 900. So, and the naming, how to name this restriction enzyme, here you can observe EcoR1. Eco comes from Escherichia coli bacteria. The R is derived from the name of the strain, and Roman numbers indicates the order in which enzyme was isolated from the strain of bacteria. So EcoR1, Eco Escherichia coli. Here you can see R is derived from the name of strain, and Roman numeral indicates the order in which enzyme is isolated from the bacteria. So, to cut your desired DNA, you require restriction enzyme. You require restriction endonuclease. There are two types. One is exo and one is endo. See, what is exo? The name only indicates. So exo removes nucleotides ends of the DNA, whereas endo makes cut at specific position within the DNA. So these enzymes require a particular sequence. It recognizes only a particular sequence. That sequence, we call it as palindromic nucleotide sequence. So what is palindromic? Observe the letters carefully. GAATTC. So if you read in reverse order also, GAATTC, correct? Same direction, GAATTC, and opposite also same, GAATTC. The number 1001. 1001, so you can read in both the ways. So same, this type of sequence we call it as palindromic sequence. And in your textbook, one more word is given, Malayalam. So if you observe this letter, same. It is a good example for palindromic sequence. So your restriction enzymes cuts only palindromic sequences. Here you can see. So this is the picture of recombinant DNA technology. Now I have a desired DNA. Just for example, I am telling you the DNA is responsible for insulin production. You can cut the DNA into many fragments with the help of restriction enzyme. Once the DNA is ready, you can observe this blue color double-stranded DNA. Once the DNA is ready, you have to insert the DNA into vector. You can see the vector is a plasmid. Now the question arises, what is vector? Vector are cloning vehicles. Vectors are plasmids. What are plasmids? These are extra chromosomal DNA present in bacteria. So plasmids are extra chromosomal DNA present in bacteria. So take this plasmid, cut the plasmid with the same restriction enzyme. So the gap is created. Now easily this blue colored desired DNA is introduced into cloning vector. And you can observe the enzyme here, the DNA ligase, which can join foreign DNA to plasmid. Again, I'll repeat. Observe this pattern. I have a desired DNA, cut desired DNA with restriction enzyme. The fragments are inserted into vector. What is vector? It is a plasmid. It is an extra chromosomal DNA present in bacteria. Cut the plasmid with the same restriction enzyme. Now you can insert this foreign DNA into vector. It is joined with the help of an enzyme DNA ligase. How it joins? It creates phosphodiester bond between 3' hydroxyl group of one end and 5' phosphate group of another end. So it is also called molecular stitcher. See, now you can observe the picture. This is the vector DNA, plasmid DNA. It has palindromic sequence. This is the desired DNA. So restriction enzyme will cut the desired DNA. You can observe. One is L shape, another one is inverted L, ulta L. So easily the vector DNA is joined with desired DNA. This is called recombinant DNA. You can see the combination. One is desired DNA, other is plasmid DNA. You are combining desired DNA with the plasmid DNA. Therefore, it is recombinant DNA. So now the recombinant DNA is ready. Any doubts? Clear? Hello? Any doubts? Trisha? Are you listening?
No, ma'am. I understand. Yes, ma'am.
Okay, what are restriction enzymes?
They are molecular scissors which cut the DNA.
Okay, where it will cut actually?
It will cut the foreign DNA.
Okay, restriction enzyme will cut the foreign DNA, but position where it will cut? There is a specific sequence. It recognizes a specific sequence. That specific sequence is called?
Sticky head.
Okay, once it cut, it releases sticky ends.
Restriction area.
Yes,
Yes, ma'am.
the sequence is
Recombinant
called,
area. That part it will cut. Palindromic sequence.
correct, palindromic sequences, it will cut. Okay, what are vectors?
Vectors. They are the Areas at which it cuts, no, ma'am.
No, no, no. I'm asking you, this is desired DNA,
It
DNA
is
cut.
through which carries the insulin,
Yes. So
on which
vector.
the insulin will be able to
Hmm.
grow.
Okay. Okay. These vectors are the molecular vehicles, actually cloning vehicles. It is present in bacteria. It is a extra chromosomal DNA. So easily your foreign DNA, your desired DNA, maybe insulin, maybe growth hormone or any other, basically your desired DNA can be inserted into this vector. So this is the recombinant DNA. And what is the function of ligase?
Ligase is to join it. It is glue.
Correct, it is molecular glue. How it joins actually?
Join the sticky ends. It will
Hmm. Yeah, it joins sticky ends by creating bond, phosphodiester bond between 3' hydroxyl group and 5' phosphate group. Okay. And once this recombinant DNA is ready, it can be easily transformed into bacterial cells. For transformation, I'll explain other techniques. So easily recombinant DNA can be inserted into bacterial cell. And we know bacteria will multiply fast. So bacteria undergoes binary fission. So one cell into two, two into four, four into eight, like this. So this is about recombinant DNA technology. So we'll get questions on restriction enzymes, cloning vectors, about foreign DNA, as well as the function of ligase, transformation, or the complete diagrammatic representation of recombinant DNA technology.